So, so it's an absolute delight to have Professor Lagace with us, who's a director of the Lipoprotein Receptor Biology Laboratory at the University of Ottawa Heart Institute, and also associate professor in the Department of Biochemistry, Microbiology, and Immunology, University of Ottawa. And Dr. Lagace obtained his PhD back in 2004, studying cellular cholesterol and phospholipid metabolism in Dr. Ridgway's lab at the Dalhousie University. And then he did a postdoctoral fellowship with Dr. Jay Horton in the Department of Molecular Genetics at University of Texas Southwestern in Dallas. This is, of course, where Brown and Goldstein, the Nobel Prize winners in cholesterol metabolism were in that cluster. Really very, very exciting in terms of the therapies that's beginning in terms of LDL chemistry. And his project was particularly timely from that point of view as well, because he was studying the secreted protein, serine protease, PCSK9, and which was identified. I think it's really actually one of the poster models of a target that's identified through genetics, and then the biochemistry that's worked out so quickly that therapy can be implemented in less than a decade, which is now used every day for our patients. And over the next 4 years, Dr. Lagace really played a leading role in terms of understanding the PCSK9 inhibition strategies on the LDL receptor and all the biology that followed. And I remember speaking with Tom at one of the CIHR Young Investigator events where he was presenting a poster on this very novel protein at that time. And I was absolutely fascinated by biology and really spent quite a bit of time, sort of really chatting on the intricacies of this pathway. And but this is before he actually decided to, return to Canada. But I certainly was very much admirer of the work, and so it was a delight, to actually meet you again here back in Ottawa. And he took up his position here in January of 2009, and he has subsequently really unraveled the cellular molecular mechanisms that control cholesterol homeostasis, and focus being on the PCSK9 biology. And so this is really, I think, is one of the current models of how therapies can be developed, sort of deriving from Mendelian randomization and, sort of the causal target, and then now protein therapies, but now really exciting RNA therapies. Now it's really being approved in Canada, starting to be used worldwide. And it's really another archetypal model of protein regulation, and can really teach us about the mechanisms of protein mechanisms of normal biology, but as well as in terms of when there's a dysfunction leading to disease. And the recent linkage, PCSK9 onto inflammation, which is going to be one of the targets of the talk, is a really exciting example of that. And there's research being supported through CIHR, NSERC, Heart and Stroke Foundation, Pfizer Aspire programs. And so this is really actually, I think, paving the way for the future of protein-based therapeutics. And so today, I'd like to welcome Dr. Lagace to give us a discussion on the topic of intertwined roles of PCSK9 in lipoprotein metabolism and inflammation. Tom?
Okay, thank you, Peter, for the very kind introduction. Thanks everyone for coming. Nice to see some people in person as well, and welcome everyone online as well. Okay, so I'm going to talk today about my favorite protein, PCSK9, and its role in binding and regulation of another protein called the LDL receptor. And there's been a lot of interest, as Peter mentioned, in PCSK9 biology because of this function of PCSK9, mainly related to us trying to find ways to inhibit its ability to bind and degrade the LDL receptor. All right, so as many people I'm sure appreciate, there's a very pivotal role of LDL in the atherosclerotic lesion development. And this starts with, in the initiation phase of an atherosclerotic lesion, the LDL particles can move from the out in the bloodstream into the subendothelial space by a process called transcytosis across an endothelial cell. And once in there, it kind of gets modified through pro-oxidant factors into what's called oxidatively modified, or ox-LDL. And instead of being taken up through mechanisms that undergo negative feedback regulation like other cholesterol homeostasis involves, the oxidized LDL gets taken up through scavenger receptors, which are not regulated by the level of cholesterol within the cell. So a cell such as a macrophage that takes up lots of oxidized LDL in this manner can become very engorged in that lipid. And so eventually becomes what's known as a foam cell. Over time, this eventually will lead to cell death, either apoptosis, which would be a good thing, or bad types of cell death, like Dr. Rayner studies, like necroptosis and necrosis. And this releases pro-inflammatory factors, which then recruit further monocyte infiltration into the subendothelial space. And it's kind of a perpetuating cycle throughout a lifetime that eventually leads to a enlarged atherosclerotic plaque development. All right, so one of the initiating factors and then contributing factors to the progression of the lesion is the LDL cholesterol. So there's lots of ways in which we try to lower LDL cholesterol in the bloodstream. What's been around for decades are the statin drugs, which will increase the LDL receptor expression in liver hepatocytes. Ezetimibe is a drug which lowers cholesterol absorption from the diet. And then the new player on the block are these PCSK9 inhibitors that have been around now for about 7 years in being taken by patients. So these are very, very potent at lowering LDL cholesterol, but they also kind of, these very important treatments show us that there's still a continuous residual risk, even when you have a very, very high level of LDL lowering, and that's coming from the inflammatory factors. So there's also a need for extra treatments in order to go after the inflammation. And so the ideal therapy would be a dual one, which targets both the lipid and the inflammation aspect. LDL, or ox-LDL, we can think of as a link between the cholesterol accumulation and the chronic inflammatory process of atherosclerosis. See, the ox-LDL can bind to these scavenger receptors on the cell surface of an immune cell. Here, this might be a macrophage or a T cell that would express this scavenger receptor, or pattern recognition receptor, as it's also known, called CD36. And this and other receptors like it can bind onto the oxidized LDL, and that one thing, it will internalize the oxidized LDL, but also triggers downstream signaling pathways, right? So the cholesterol itself can accumulate to such a level within the endocytic pathway that can form cholesterol crystals, which cause disruption of lysosomes and activation of the NLRP3 inflammasome. This can also cause mitochondrial stress, ROS production, as well as activation of transcription factor pathways such as NF-kappa B, which will increase the production of inflammatory cytokines. So ox-LDL through this pathway is what's known as a danger-associated molecular pattern, or DAMP. So it has ability to activate these immune system pathways that are very pro-inflammatory. All right. And we can think of oxidized LDL as sort of, again, a circular perpetuating cycle, because as more of these pro-oxidant pathways are activated, more and more of the LDL that infiltrates into the subendothelial space will become oxidized LDL. So it's a bit of a vicious cycle. All right, so a lot of what we know about LDL metabolism comes from the study of single gene disorders. So the liver is the main producer of circulating cholesterol, and it produces a lipoprotein called very low density lipoprotein, or VLDL. It has a molecule called ApoB100 attached to it. And then as it circulates through the circulation, undergoes enzymatic processes such as lipolysis, remodeling. Eventually, some of the triglycerides get removed, and it becomes more of a cholesterol dense particle called intermediate density lipoprotein, or IDL. At this point, it could acquire a ligand called ApoE that can bind to the LDL receptor. and be internalized into the liver, or it stays in the circulation and becomes a very cholesterol-rich particle called LDL, or low-density lipoprotein. At this stage, the ApoB100 has undergone some conformational changes in which it can now be recognized by the LDL receptor, and the LDL particle can be internalized into the liver, where the lipids are repackaged for secretion into VLDL or used for cellular metabolism. So the single gene disorders of LDL metabolism predictably are involved in the genes that regulate this process, mainly the LDL receptor gene itself, and also the gene for ApoB100, the ligand of the LDL receptor. In 2003, through genetic studies of patients that had hypercholesterolemia, but didn't have gene defects in the LDL receptor or ApoB, they discovered a new gene involved in the disease autosomal dominant hypercholesterolemia known as PCSK9. So this happened in 2003. So what is PCSK9? It stands for proprotein convertase subtilisin kexin type 9. It's a serine protease, and as its name suggests, it's the 9th member of this family of proprotein convertases that has been discovered. It's a secreted soluble protein with highest expression in the liver, but it's also expressed in multiple other tissues. It undergoes an autocatalytic processing in the endoplasmic reticulum, where it cuts itself. And then, as with other PC enzymes other PC enzymes function in the limited proteolysis of substrates in the secretory pathway or in the extracellular compartment. However, PCSK9 doesn't seem to have any other proteolytic substrate other than itself in this autocatalytic mechanism. A lot of what we know about PCSK9 has come through functional studies of mutations that affect its activity. These are either loss of function mutations, and I'm just listing a few of them here. They're found throughout the protein's sequence, and these are associated with a lowering of LDL cholesterol in people, and then there's a decrease in the cardiovascular heart disease risk. Alternatively, there's gain of function mutations that can cause an increase in LDL cholesterol and an increase in CHD risk. And I would say most of what we know about PCSK9 function and validation of a lot of our studies has come through the study of these gain of function mutations, because they have a very big effect on circulating LDL cholesterol, whereas the loss of function mutations are often more prevalent in human populations, but have a smaller effect size on LDL cholesterol. So they're more difficult to functionally characterize. This has been a bit of a double-edged sword, because in the early days, there was a lot of people being thrown off by this S127R mutation in PCSK9, because now we know this greatly decreases the processing and secretion of the PCSK9, but it still does get secreted, still does get out in the circulation. But in the early days, before the advent of really good antibodies to PCSK9, we thought that S127R was not secreted at all. And in fact, there was also some studies suggesting that another mutation, D374Y, was not secreted. So everyone really thought in the early days that PCSK9 must be acting intracellularly within the secretory pathway. But our first break came with our making some antibodies to PCSK9 in mice that were really good at detecting the endogenous protein. And we're able to see that PCSK9 was actually secreted very efficiently in hepatic cells. And this is a standard pulse chase analysis. You can see the precursor form of the PCSK9 and then the cleaved mature form. And then this doesn't build up in the cells over time, but rather gets secreted and recovered in the culture media. And even after 4 hours, there's very little of the PCSK9 left within the cell. It's an idea that maybe it's working from the outside. We added the PCSK9 back to the cells, and we saw that it was taken up very well into mouse embryonic fibroblasts that were expressing the LDL receptor, but not mouse embryonic fibroblasts that were derived from LDL receptor knockout mice. So this was our first indication that perhaps PCSK9 was able to bind to the LDL receptor, but we didn't know whether this was direct or indirect, right? So here's where we turn to some of the gain of function mutations. We looked through different ones to see if any of them increased this activity. And then we also did a direct binding assay where we took purified LDL receptors, ran them out on a non-reducing SCS page gel, and then blotted it directly with the PCSK9 protein. You can see the binding of a radioactive PCSK9 to the LDL receptor using wild-type PCSK9. And then there's a dramatic increase in the binding when you use the gain of function D374Y, right? So now, even though we're doing a test in vitro, it really is validated through this study of these gain-of-function mutations, because now we know we're definitely on the right track, because this is something that's causing hypercholesterolemia in human patients. We developed an ELISA assay, the first ELISA to detect how much PCSK9 there was in the human plasma. And then we used physiological concentrations of PCSK9 introduced into mice through a constant infusion in order to show that PCSK9 could act within the circulation at a physiological level. So if you inject or infuse 32 micrograms of PCSK9 per hour, this comes to, it ends up with about 400 nanograms per mil of PCSK9 at steady state, which is about the level that's found in human plasma. You can see almost the complete destruction of the LDL receptors in hepatocytes under these conditions. So we knew that the PCSK9 now was acting as a secreted factor. It was binding directly to the LDL receptor. Some other studies we did showed that PCSK9 didn't require its catalytic activity, its inherent catalytic activity, but that rather it was acting as a binding chaperone. So this informed the pharmaceutical approach of inhibiting PCSK9 through the use of injectable monoclonal antibodies that recognize the part of the protein that binds to the LDL receptor and basically jam up that interaction. So these were quickly developed by the pharmaceutical industry and became available for use in patients in 2016. Crystal structure of the very first one that was developed by Amgen. This is now called Repatha, the trade name, or volocumab, which is the name of the monoclonal antibody. You can see how it basically binds to PCSK9 at a region in a part of the PCSK9 catalytic domain that binds to the LDL receptor EGFA domain. And that antibody binding there with very high affinity prevents the LDL receptor from binding, or PCSK9 from binding to the LDL receptor. So now there's two of these that have been approved for use in patients, one from Amgen, also from Regeneron. And the problem is that there's these antibody treatments are very effective, up to 70% lowering of LDL cholesterol, but they're very expensive as well. And so the amount of people needed to treat to get the reductions that you need in cardiovascular events has a very exorbitant cost on the healthcare system. So there's always a need for better and more cost-effective ways to inhibit PCSK9. There's an siRNA that targets PCSK9 in the liver called inclisiran that's been developed by Novartis, and this was approved in 2020. And we're still waiting on clinical data on the efficacy of this treatment. However, it's very good at lowering LDL cholesterol, not quite as good as the antibodies. It's about 50% lowering. But this is only targeting the PCSK9 produced in the liver. And that may be something important because we now know the PCSK9 is also being produced in other cell types, including cell types within the atherosclerotic plaque itself, right? There's also a little bit more out there, I'd say, ideas about targeting PCSK9, such as vaccination against the protein itself or CRISPR gene editing to remove it from the genome. But these are really, I would say, very risky at this point because we still don't understand a lot about PCSK9 biology. And I, for one, wouldn't be first in line for one of these treatments. All right, so a lot of what we know, as Peter mentioned, about cholesterol homeostasis and the LDL receptor pathway has come from the lab of Drs. Joseph Goldstein and Michael Brown at UT Southwestern in Dallas. So they won the Nobel Prize for deciphering the LDL receptor pathway back in the 1980s, which led to helped lead to the development of the statin drugs. They also came up with this cellular pathway, deciphering all the different parts of it, called the SREBP pathway. This is a gene transcription pathway that regulates a whole host of genes that are involved in cholesterol metabolism. So what happens is the SREBP protein is a membrane-bound transcription factor bound within the endoplasmic reticulum. And when sterol levels are low, or cholesterol levels are below a certain threshold, this protein called INSIG dissociates from a protein called SCAP, which then can bind to the SREBP and transport it in vesicles transport to the Golgi apparatus, where it encounters a protease called site 1 protease that clips it in a luminal domain. And then a second protease called site 2 protease clips it at a transmembrane region. And this releases a basic helix loop helix transcription factor domain that traffics to the nucleus and binds to a sterile regulatory element within target genes. These target genes include the LDL receptor as well as cholesterol biosynthetic genes. So in response to low cholesterol levels, there will be an upregulation of the gene expression of these proteins. And conversely, if cholesterol levels are high or elevated, INSIG gets stabilized by the cholesterol, binds to the SCAP, and anchors this complex in the endoplasmic reticulum so that it no longer can traffic and become activated. So there's multiple genes regulated by the SREBPs. There's two different types. SREBP2 mainly regulates genes involved in cholesterol metabolism, whereas SREBP1C mainly is involved in genes regulating the production of fatty acids and triglycerides within the liver. Interestingly, there's two genes that are regulated by SREBPs that are negative regulators of the pathway. Right? So INSIG, we saw on the last slide, is a protein that can bind to SCAP and anchor it in the endoplasmic reticulum. But INSIG is regulated ultimately by cholesterol. If cholesterol levels are low, INSIG is degraded so that the SCAP SREBP complex can traffic to the Golgi. PCSK9 is the other one. It's regulated by SREBP2. So this has always been a bit of a conundrum. When you think about why do we have PCSK9? Why is it regulated by the SREBP pathway? Well, for one thing, we know that PCSK9 blunts statin efficacy because of the way that statins work. So statins are direct inhibitors of HMG-CoA reductase, the rate-limiting enzyme in cholesterol biosynthesis. This in turn, this decrease in cholesterol biosynthesis leads to the cell becoming depleted for cholesterol, meaning there's an upregulation of the SREBP2 transcription factor. This increases LDL receptor mRNA expression and LDL receptor protein expression at the cell surface. So this leads to increased LDL uptake in cells. So the efficacy of the statin drugs is a secondary effect to its main effect, which is the inhibition of HMG-CoA reductase. But we know that PCSK9 is also an SREBP2 target. So it seems very counterintuitive that at the same time when the cell is deprived of cholesterol, needs the LDL receptor expression to take up LDL cholesterol, it's also making something that's going to degrade the LDL receptor. So our hypothesis, an overarching hypothesis, which has kind of guided our work, is that PCSK9 activity must be regulated at some level by cholesterol. And since it is a secreted factor, it's likely that it could be regulated by lipoprotein cholesterol, which in humans is mainly carried in LDL. Show in a paper published in 2013 that a lot of the PCSK9 in blood plasma is bound to LDL particles. At least 30 to 40% of PCSK9 is bound to LDL particles in individuals with normal lipidemic levels, right? And through immunobinding, LDL immunodepletion, we're able to show that PCSK9 is binding directly to ApoB, and that's pulled down in an immunoprecipitation with ApoB. And this is further evidenced by in vitro binding studies in which PCSK9 binding to LDL is shown to be very specific, saturable, and consistent with a one-site binding model. So these are gel shift assays where we take labeled PCSK9, bind it to LDL, and then separate the reaction on an agarose gel. And we can see, we can quantify the bound PCSK9, and that is separated from the free, that which is free. So that LDL can inhibit the PCSK9 LDL receptor interaction, at least in cultured cells. When PCSK9 is pre-incubated with LDL and then the mixture is given to HUH7 cells, the LDL will inhibit the ability of PCSK9 to cause the degradation of the LDL receptor. You can see here. And also when you give cells PCSK9 in the presence of LDL, its ability to bind to cell surface LDL receptors is blunted somewhat. So this is just showing that the PCSK9 uptake into cells is being inhibited by increasing concentrations of LDL. And this is LDL in a physiological range found in human plasma. And importantly, we showed that PCSK9 also does the same thing to when the LDL receptor is missing a part that binds to the LDL particle itself, right? So the LDL particle is not directly affecting the binding of PCSK9 to LDL receptor through a direct blocking effect, but rather we think it's having some effect on the structure of PCSK9 and its ability to form its binding interaction with the LDL receptor. And more of this evidence has come from our mapping studies. So this is a schematic of PCSK9, the proform. And then, as I mentioned, it gets cleaved, it cleaves itself. The prodomain reattaches to the catalytic domain, and this covers up the catalytic site, prevents any substrate from coming in. And the prodomain is also a very important chaperone for the protein. You can't remove the prodomain from PCSK9 and still get the protein secreted. And even after the protein secreted, you can't artificially remove the protein or the prodomain, or else the rest of the protein just becomes aggregated, unfolds, and falls out of solution. So the prodomain is a very important chaperone. Interestingly, it has an N-terminal disordered region, which is missing in all the crystal structures of PCSK9 that are in the protein database, over 30 in total, are all missing structure information for this N-terminal disordered region in the prodomain. And we found early on that if you remove this disordered region, the binding to the LDL receptor becomes better. So PCSK9 binds with higher affinity to the LDL receptor when this disordered region is removed from the protein, even though it is very far removed from the actual binding site to the LDL receptor EGFA domain. So we think it's some type of a autoinhibitory domain that's having an allosteric regulatory effect on the LDL receptor binding interaction. Now, interestingly, we found that it's also required for LDL binding. If you remove this region from the protein, it no longer is able to bind to LDL particles. So it has a dual role in regulating PCSK9 binding to the LDL receptor and to LDL particles. So looking at this disordered region, you notice jumps right out at you at first that there's these two areas. There's an acidic domain and a hydrophobic region. And also that there's very good conservation of this region between different vertebrates, but not between vertebrate and invertebrate PCSK9. So this kind of tells you this might have an important evolutionary role in PCSK9 function. So we altered these two regions in PCSK9 through a nested deletion analysis. So either we took out the acidic domain, or we replaced the hydrophobic domain with a disordered linker so that we didn't alter the spacing. And we found that in each case, there was a big decrease in the LDL binding ability of PCSK9, suggesting some type of a functional overlap between these two regions. Then if you take the sequence and you model it, there's a secondary structure prediction method called CyPred. It actually predicts that this would form a helix that overlaps both the acidic and the hydrophobic regions. And then when you place this on a helical wheel model, you can see that it lines up a hydrophobic face and then a more hydrophilic side as well. So this is what we call an amphipathic helix. Often these are involved in binding to lipids, binding to lipid membranes. A lot of apolipoproteins, like apolipoprotein E, for instance, have amphipathic helices that allow it to bind to lipoproteins. But this one's quite small. But just the same, it could be formed in the presence of lipids. You can't see it, but I can see it.
Probably need to stop sharing and rejoin it. Yeah. So this is your presentation.
talked about. Suspense. What happens when we alter the helix? All right, so we put a proline residue, which is a type of amino acid that doesn't naturally form into an alpha helix because of different conformational constraints. And if we put this helix breaking A44P mutation in PCSK9, we found this greatly lowered the binding affinity to the LDL particle, not quite as dramatically as removing the N-terminus completely. It lowered the binding affinity about 5-fold. And it didn't have any effect on the ability of PCSK9 to bind to the LDL receptor or to mediate LDL receptor degradation. So this N-terminal helix is critical to LDL binding, but not to binding to the LDL receptor. And interestingly, it contains a loss of function mutation, which is one of the very first ones discovered in PCSK9. This is found in about 3% of people of European Caucasian descent, and is atheroprotective. That was found back in 2006, one of the first ones, and it's very prevalent and often used in genetic studies of PCSK9, and even as a surrogate for cholesterol-lowering therapies because it's so prevalent. So we found that the R46L, if you put it on a helical wheel model again, it increases the hydrophobic phase of the helix. And it's also predicted in a thing called a helical projection. It's predicted to increase side chain interactions to further stabilize the helix. So we kind of thought, oh, this is going to increase LDL affinity because the helix is so important. But we were very disappointed to see that the LDL binding affinity didn't really change between wild type and R46L. And I think this really goes back to what I talked about in the beginning. It's really, really tough to functionally characterize these loss of function mutations because R46L is associated with about a 10% decrease in LDL cholesterol. So a very mild effect compared to some of the gain of function mutations, which have a big, big effect, 3 or 4-fold increase in LDL cholesterol. So we really have a hard time to model these. So structural studies can have a really important role in this way. So we just looked at the peptides themselves of the N-terminus containing either the wild type peptide or containing this R46L substitution. And this is a circular dichroism experiment where you can tell whether the peptide adopts helical structure in the presence of a detergent micelle. So this is a hydrophobic environment. So a DPC detergent micelle is added, and you can see that there's a characteristic maxima and minima that are of alpha helical formation in this type of an experiment. And we can see that this is greatly enhanced by the presence of the R46L mutation or substitution within the peptide. And this correlates to about an 8-fold increased affinity of the R46L containing peptide for the DPC micelle. So we really think that the R46L loss of function in some way might involve an increased helix propensity in the N-terminus of the PCSK9 protein that in some way leads to a lowered function of that protein in humans. Time to see if PCSK9 was binding to lipids through this amphipathic helix, since it has this hydrophobic phase. But as I mentioned, it's quite a small region. So we next turn to computational modeling, using a process called molecular dynamic simulation, to try and see if we could see what this N-terminal helix might be interacting with. And we did this with a collaborator, Ariela Vergara-Jacques, who is in Chile. And she was introduced to us through Emilio Alarcon, one of my colleagues here at the Heart Institute. And she did this really great work to come up with this model of PCSK9, the full-length PCSK9, including that disordered N-terminal region. She even found a second helix that we're also interested in pursuing further upstream. But this is the main one that we were talking about before. And her model predicts that it's actually forming an intramolecular interaction with a region of the C-terminus of PCSK9. And there's several gain-of-function mutations that are found at the predicted interface. So when we tested those gain-of-function mutations to see if there had any effect on LDL binding, lo and behold, there was a huge decrease in the LDL binding. This one, R496W, completely abolish the ability of PCSK9 to bind to LDL particles in vitro. And none of these mutations seem to have any effect on the ability of PCSK9 to bind to the LDL receptor. And we did some more detailed experiments, I'm not showing here for the paper, showing that the purified R496W protein was equal to wild-type PCSK9 in its binding affinity to the LDL receptor and also its ability to mediate LDL receptor degradation. So it's very likely, given that the gain of function of these that's associated with these mutations is related to its inability to bind to LDL particles. So we next wanted to look at, as I mentioned, there's these gain of function mutations spread throughout the protein. So looking at the structure of PCSK9, you see this is the part that we identified in this region of the C-terminal domain. And if you line it up along the same molecular plane, you see there's this other grouping of gain of function mutations in the prodomain that are really along the same line. So we wanted to see if any of these also affected LDL binding. And sure enough, they did, including that first one that was discovered in FH patients way back in 2003, S127R, which has been very enigmatic throughout all this time. Hasn't really been able to be deciphered how it's causing a gain of function. We found that this was very, very detrimental to binding to LDL particles. And another one right by, a D129G, and we're now looking at other mutations at this D129 site as well. Right, so what happens then if you have one of these gain of function mutations that inhibit LDL binding? What happens in terms of binding to the LDL receptor? Well, we can see that a protein that has a D374Y, so it binds really well to the LDL receptor, as we've shown previously, this is inhibited by LDL in its ability to bind to LDL receptor and be taken up into the cells. Whereas if the protein contains this S127R mutation, it's no longer inhibited by LDL. So we have to test this in vivo in a physiological model, but at least in cell culture, it looks like these gain of functions are relieving an inhibitory effect of LDL, right? These two sites, we can kind of envision a model where PCSK9 is basically docking onto an LDL particle involving these two regions, which are far removed from each other, but on the same, roughly the same molecular plane of the molecule. And this is quite far away, again, from the LDL receptor binding site, which is over here in the catalytic domain. Through this interaction and through this binding to the LDL particle, there's conformational changes in the LDL in the PCSK9 that affect its ability to bind to LDL receptor. But what these are, we don't know yet. And we're still going to keep studying this allosteric mechanism, because we think that by binding to an LDL particle and forming this helix, that the PCSK9 is locked into an auto-inhibited conformation. And that raises an exciting possibility that you could come up with a small molecule inhibitor that could bind to a part of PCSK9 and mimic this inhibitory effect of LDL. All right, so in summary, 30–40% of PCSK9 in human plasma is bound to LDL particles. So this is a very significant amount of PCSK9 that's bound to LDL. And we showed that there's numerous gain of function mutations associated with hypercholesterolemia that inhibit or prevent LDL binding. I'm showing a list here, and we're continuing to find more and more and more of these mutations along PCSK9 as we keep testing them. And importantly, these mutations identify regions in PCSK9 that are critical to LDL association, and this will help us in our further studies to hopefully identify small molecule inhibitors of PCSK9 through this mechanism. We've also been looking at whether PCSK9 how it can bind to different classes of ApoB containing lipoproteins. You recall the liver secretes the VLDL particle that then becomes the IDL, or intermediate density lipoprotein, and then becomes the low density lipoprotein. Well, where does PCSK9 bind to ApoB100? Is it on the VLDL right away, or does it, like the LDL receptor, does it have to wait until it becomes a certain cholesterol density in the metabolism pathway? So we found through in vitro studies that PCSK9 binds quite well to IDL, intermediate density lipoprotein. You can see through this gel shift assay. Of course, it binds to LDL, and we don't detect any binding of PCSK9 to VLDL, right? So this sets up a sort of a scenario where maybe PCSK9 is able to sense the lipoprotein remodeling that occurs in the intravascular compartment. So as the liver secretes a lipoprotein, very triglyceride-rich lipoprotein VLDL, and as this gets hydrolyzed through processes like lipoprotein lipase, hepatic lipase, and then remodeling through cholesterol ester transfer protein, the triglycerides are hydrolyzed to fatty acids that are being delivered to tissue cells for use for fuel. The VLDL is being metabolized to the short-lived IDL and finally to LDL. Well, at this stage of its maturation, it can acquire an apolipoprotein E, an exchangeable apolipoprotein. This is also a ligand of the LDL receptor. So the LDL receptor could clear the lipoprotein at this early stage. And that would deprive perhaps these peripheral tissues of these fatty acids that are needed for fuel. And this could be important under certain conditions, such as a prolonged fasting or something like that. But with a modern Western diet, we really don't need a mechanism like this that would protect our lipoprotein levels within our circulating plasma. So as the lipoprotein then becomes metabolized, eventually the PCSK9 can bind to the apolipoprotein B100, and that inhibits its ability to bind to the LDL receptor. And of course, these gain-of-function mutations would not be subject to this inhibition because they're not able to bind to the LDL. So they remain more active. They remain free and active. So as we know, PCSK9 is regulated by SREBP2, which also regulates the LDL receptor. But it's also known that PCSK9 gene transcription is regulated by HNF1 alpha, which is a transcription factor which is activated in the liver in response to inflammatory stimuli. So PCSK9 secretion goes up after a meal, and that's mainly through the SREBP2, but it's also increased, its mRNA at least is increased by inflammation through HNF1 alpha. And it's been shown that PCSK9 increases its mRNA expression is stimulated by pro-inflammatory stimuli such as lipopolysaccharide or LPS or OX-LDL in vascular smooth muscle cells, endothelial cells, macrophages. These can all produce and secrete PCSK9 as well. Although the liver is still the main source of circulating PCSK9. And it's also been shown that this source is also increased in response to LPS in mice, at least through the mRNA expression level. And what's really exciting is all the plethora of papers that are coming out on the links between PCSK9 and the and immunity, inflammatory processes. Some of these are very high impact papers showing the involvement of PCSK9 in these processes, such as septic shock syndrome. There's a better outcome in people that have loss of function mutations in PCSK9, and also PCSK9 inhibitors are having efficacy in clinical trials of septic shock. There was a paper came out in Nature that PCSK9 was having an immune checkpoint effect in a cancer model in mice. And importantly, this was done as well in an LDL receptor knockout mice. The same effects were present. So clearly this is an LDL receptor independent effect. Other studies have looked at PCSK9 as a novel immunological factor in oxidized LDL-induced immune cell maturation, activation, processes like thrombosis, all kinds of different things are being affected by PCSK9, mostly in cellular studies, but also some in vivo ones, and often done in LDL receptor knockout mice. This is one that just came out pretty exciting that they used based on these types of interactions of PCSK9 with the immune system. They tested PCSK9 inhibitors in patients that had severe SARS-CoV-2 infections. And they just published this in JACC, I think last week, that there was a 30% decrease in deaths and the need to intubate these patients in the ones that received the PCSK9 inhibitors compared to placebo. There was also a 30% decrease in the pro-inflammatory cytokine IL-6. So something about these inhibitors is really having an effect on these types of acute immune responses. So we're kind of pulled into this through some studies done by Ikuosho, who's here, who's looking at a form of PCSK9 that's found in the blood that's cleaved by furin. this is one of the proprotein convertases, also known as PCSK9. So furin is a transmembrane bound protease expressed in the secretory pathway and also at the cell surface. It cleaves PCSK9 at a part in the catalytic domain, and about 10 to 40% of PCSK9 in human plasma appears to be in this furin cleaved form, this truncated form. It's thought to be inactive, and I put this in quotation marks because we really don't know. It might have another activity, but it's definitely defective for LDL receptor binding and degradation. So since the furin cleavage site is right in a part that overlaps the overall binding interface to the LDL receptor. So, and it's also been known that truncated form of PCSK9 in patients receiving the PCSK9 inhibitory antibody evolocumab is decreased. And this is perhaps not surprising because these antibodies bind to the same region. This is the region that binds to the LDL receptor. And we confirmed this looking at the inhibitory antibody aliricumab. When you incubate PCSK9, pre-incubate with aliricumab, it's now resistant to furin proteolysis. What happens to furin cleaved PCSK9 in terms of its binding to LDL particles, right? It can't bind to the LDL receptor, but it actually binds better to LDL particles. You can see here. So this is the uncleaved PCSK9 and FC is the furin cleaved PCSK9. And the binding of PCSK9 to oxidized LDL is less, but the binding of the furin cleaved one to the oxidized LDL is greatly augmented, about 12-fold increase in the binding of the furin cleaved PCSK9 to ox LDL. And mechanistically, it could be involved. We don't know whether it's infecting the binding to the ApoB100 moiety of LDL. It could also be through a lipid interaction, because we found that in liposome binding assays in which you just incubate purified PCSK9, either the uncleaved one or the furin cleaved one with liposomes, and then you do a centrifugation to collect a pellet that contains the liposomes. If protein's bound, it's found in the pellet, and if it's not bound, it's found in the supernatant. So you can see that there's a great increase in the binding of the furin cleaved PCSK9 to the liposomes compared to the uncleaved one. And these are liposomes that have a phosphatidylcholine sphingomyelin to cholesterol ratio of 60-20-20 mole percentage. This is roughly the percentage that's found in the outer membrane of a lipoprotein particle. And interestingly, it's also found in cholesterol-rich lipid rafts, such as caveoli, that are signaling platforms in immune cells. So it's possible that PCSK9 has this sort of attraction, or the furin cleaved one at least, to these types of lipid membranes. From Natasha, from Aaron Mulvihill's lab, we looked at what happens when you treat mice, wild-type mice, female mice in this case, with LPS. So after 24-hour LPS treatment, we saw an increase in the plasma concentration of PCSK9 as measured by immunoprecipitation immunoblot, not just the full-length PCSK9, but also the furin cleaved form of PCSK9 goes up in this inflammatory condition. And Ikuosho added the different forms of PCSK9 to THP1 macrophages, and I found that in the presence of ox-LDL, the furin cleaved PCSK9 uptake into the M1 pro-inflammatory macrophages was greatly increased compared to the uncleaved protein. PCSK9 then augment the ability of ox-LDL to bind to scavenger receptors that are found on the surface of these immune cells, such as CD36 or lectin or LOX1. So these are types of scavenger receptors. So in this experiment, Ikuosho overexpressed these receptors in HEC293 cells, gave them either a protein in which most of it's uncleaved or one in which most of it's furin cleaved, gave that to cells to see which ones would be taken up better. And in the presence of LOX1 or CD36, there's a much greater increase of the furin cleaved one uptake into cells. And this was further augmented in the presence of ox-LDL. Either helping the ox-LDL interact with these receptors or vice versa. and also interacting directly by itself with these receptors, and it might be acting as some type of a pro-inflammatory factor in that manner. So this has led us to the idea that furin proteolysis of PCSK9 could be converting it to one of these danger-associated molecular patterns, a DAMP, like OX-LDL is. So the furin proteolysis of PCSK9, in the first case, allow it to bind to these oxidized LDL particles, and that would augment the interactions with these pro-inflammatory signaling receptors, such as CD36. And then perhaps the PCSK9 lipid binding properties are also involved in movement of this complex to these signaling platforms, these caveoli in these immune cells. And then this in turn can act on those downstream pro-inflammatory pathways. So in summary then, furin cleaved PCSK9 is increased under inflammatory conditions. Furin proteolysis of PCSK9 improves its OX-LDL and scavenger receptor interactions in cultured cell models. This could in turn promote downstream inflammatory pathways in immune cells. And blockage of the furin proteolysis could in part then explain anti-inflammatory effects of the therapeutic PCSK9 inhibitory monoclonal antibodies such as liraglutide. These clinical studies point to a protective effect of these PCSK9 inhibitory monoclonal antibodies in acute inflammatory conditions such as sepsis, myocardial infarction, and COVID-19. So perhaps there's a new way in which to use these inhibitors. And we should continue to study these mechanisms because we might come up with better ways to target them specifically rather than rely on these big bulky antibodies that are very expensive. Directions. So some of the main points we found is that LDL binding locks in a cryptic autoinhibited conformation of PCSK9. This involves an intramolecular interaction between the prodomain N-terminus and a C-terminal region of the protein. We have evidence that LDL binding exerts negative feedback control on PCSK9 activity through raising, as the LDL particles are elevated in plasma through the action of PCSK9 on the LDL receptor. They then are able to bind to the PCSK9 and knock down its activity. So to further test this, we need to make some AAV vectors that will express the gain of function mutants that we've determined have no effect on LDL receptor binding, and then test them in mice in dyslipidemic mouse models. And then examine the resulting effects on lipoprotein metabolism and atherogenesis in these models. And finally, proteolysis of PCSK9 by furin improves a lipid binding function, which may underlie its increased ability to bind to oxidized LDL and mediate oxidized LDL pro-inflammatory interactions with scavenger receptors. So we want to identify what the lipid binding motif is, and through structural modeling, and also hopefully finding some mutants like we've done in the past that sort of reveal this activity and help us hone in on the important parts of the protein that are involved. We want to continue to look at inflammatory pathways in cell culture, such as using smooth muscle cells, macrophages. These are important cell types that are found in atherosclerotic lesions, and especially looking at the scavenger receptors LOX1 and CD36, which we have found through looking at various ones are the ones that seem to have the biggest effect in terms of binding to PCSK9, or the furin cleaved PCSK9. And then finally, to express AAV vectors with furin resistant PCSK9 and then put them into mice to see if there's an effect on atherogenesis. We predict then that these furin resistant PCSK9 mutants would be less harmful because they don't become this pro-inflammatory form of PCSK9. And finally, in these animal studies, we want to continue to look at perhaps potential sex differences that are related to PCSK9 function. It's been known for a long time there's prevalent sex differences in cardiovascular disease. And specifically in terms of PCSK9, it's been shown that circulating PCSK9 levels are higher in women than in men, that postmenopausal women have higher circulating PCSK9 than premenopausal women, and that there's an inverse relationship between PCSK9 and estrogen in premenopausal women. So there's some type of a role of estrogen on the regulation of PCSK9 transcription and PCSK9 expression. And there's been some discordance in the different studies that have looked at PCSK9 inhibitors, but several studies have reported that these inhibitors are less effective at LDL cholesterol lowering in women compared to men. So we're really interested in whether estrogen has some type of post-translational modification on circulating PCSK9, looking at human plasma samples from women and men. And we're specifically honing in on a couple of post-translational modifications that are found in the prodomain N-terminus. One is a phosphorylation of a serine residue, and the second is a sulfation of a tyrosine residue right within that helical region. And we think this might be involved in the protein-protein interaction between the prodomain and its intramolecular targets. So we're trying to develop, we'd hope to develop an ELISA assay that can measure tyrosine sulfation in different plasma samples. As well, we want to examine this in male and female mouse models. All right, and so with that, I'm going to finish up. And I want to especially thank members of my lab, many of whom are here. Tanya, who's my lab manager, research assistant, who's been really instrumental in all these studies from day one. And Ikuosho, a PhD student, who's really spearheaded our foray into inflammation and the role of furin-cleaved PCSK9 in its function. And I want to especially highlight two past lab members, Samantha Sarkar and Angela Matias, who were instrumental in all that work on the gain-of-function mutations and their effect on LDL binding. And with that, I'd be happy to take any questions.
Great. It's a fantastic talk, and it's really the initial work in terms of painstaking, sort of unpacking of exactly, so the domains of how PCSK9 works, and the surprise in terms of the interaction with the LDL itself, in addition to the receptor. And but I think there's a whole story on the inflammation aspect that's beyond just the LOX receptor. Yeah. So maybe we can entertain some questions, from the team here in the room. Yeah. So start with Katie and the others. So feel free to use the microphone.
I can do that. I can do that. So this is great, Tom, and obviously really cool work. And one thing that strikes me when you talk about the damp aspect of PCSK9 and then potentially the binding of LDL and that hypothesis, a lot of damps are quite non-specific, in the sense that they don't, like, the cell doesn't care where they came from or what they are. They're so have you investigated like what else the furin-cleaved PCSK9 might be binding in addition to LDL and ox-LDL, like just a general proteomic something or other, or what? That's just I guess we'll start there.
Yeah, no, it's a good idea. It's actually really tricky to work with because it is sticky, and that's sort of the one of the aspects of it. But I think that when you think about a damp, it can't be something like LDL. It can't be something like PCSK9 because they're ubiquitous. They're all over the place, right? So there has to be a conversion process. Furin is highly upregulated in cardiovascular disease and inflammation. So that might be the process that converts it. It might be kind of short-lived because it is tricky to, like, we can't purify it. We can't, as soon as you remove that part from it, you almost have to use it right away. And then it seems to do these things. So I think a proteomic approach would be a really good idea. It might be a real soup of things that come out. But yeah, I think that could be something to look at. We did find, for instance, though, that it didn't bind well to TLR4. It didn't bind to SRB1, so it wasn't totally
not everywhere.
Anything.
Yeah. Okay.
But we do find like non-specific binding to cells is much higher than the full length one, that type of thing. So it's a bit.
And then along those lines, where is furin specifically upregulated? So it's cell surface, right? It's expressed, it's anchored. Yeah. So is it like, sorry, go ahead, answer.
No, it could be, that's a big question we're wondering. Is it acting on circulating PCSK9?
Exactly,
It could be liver-derived PCSK9. We actually saw at the
local.
CLC, it was a talk by Bernardo Trigati's group showing that liver-derived PCSK9 was driving a pro-inflammatory response in their mouse model. So that would suspect that it's cell surface furin that's acting on the PCSK9. It could be all the way back in the liver. It could be that's the part that gets that pro-inflammatory signal and then secretes this damp right into the circulation, or it could be furin at the lesion site.
or any site of inflammation, like if you
Yeah.
parallel it with infection or injury or something like that, is it the same? Yeah.
So some specific knockouts could be
Yeah.
really formative. There's liver specific furin knockouts
Yeah.
and other knockouts.
Cool.
So it could be good.
Okay.
Yeah.
Other questions? Let me see. So online Quang Chan's just comment. Just greatly appreciate a clear explanation of how PCS functions, you know, for in the clinical setting. So actually, I want to just follow up on that comment that Katie made. You know, so we actually look at the furin. So furin is actually very important in terms of the SARS-CoV-2 infection, right? You know, because the furin is actually the cell surface interacts with ACE2 receptor and actually processes the SARS-CoV-2 virus. And the furin is actually present in a lot of vasculature, you know, because SARS, or I guess, you know, when you actually look at COVID infection, the way I think it is actually, it's a vascular inflammation process, you know, so that's why I guess thrombosis and all the rest. So, so it makes me think that a lot of action is probably actually in the vasculature, except it's very acute, you know, so it's not the chronic, you know, sort of the LOX1, you know, type of mechanism, but actually, you know, it's related to danger signal. And so the thing that the furin and also it looks like a PCSK9, both of these seem to actually facilitate a lot of intravascular uptake of targets, you know, because that's kind of the function of these molecules, and that's kind of the way I'm thinking about it. And there are very, very specific kind of a high probability danger signals. You know, we test these, there's a whole battery of these, right? You know, we actually test these, you know, our model systems. And so makes me wonder that it's, you know, it's not a whole gemisch of danger signals, you know, that has equal opportunity, right? You know, there are these very, very high probability danger signals. So I wondered whether, in fact, this is a potential a facilitation process, you know, because it's actually internalization of these danger signals that activates, right, all the downstream.
So that's where we're really interested in the lipid binding function. I think could be the key thing because the ox-LDL, you know, might not be directed to that caveoli or the lipid raft, the signaling platform that it needs to be in order to get there. So the PCSK9 could be the key thing that sort of binds to lipids, binds especially to lipids enriched in sphingomyelin cholesterol, because it has this ability through binding to lipoproteins, but that could be the same process to bind to a lipid raft or to a membrane enriched, a domain enriched in cholesterol. So that's why I really think it's important to identify that lipid binding function and what parts of the protein are involved. And that might be the key thing that it's doing is, like you said, bringing those DAMPs to the actual, you know, target site.
And that's why the inhibitors, right, you know, are effective, right, you know, because it could potentially very much attenuate that process. So this really very, very I think that's a really exciting kind of a whole new opportunity. Other questions or comments? Okay, so I think this is really exciting area, you know, kind of bringing some of the fundamental biology, you know, into addressing some of the key problems that we all face, you know. So I think unpacking this is going to be very exciting, especially, you know, there are so many tools available. And I think even on the day one, I say that the Celestial Committee cannot actually have created the PCSK9 only for the LDL receptor. It must have other function.
It looks that way.
That's being opened up at the moment. Thank you so much. So thank you so much, and thanks everybody for joining us today, and also those online. And again, thanks to Tom for great work. Take care. Bye bye.
All right.